Hi everyone, Dr. Hinkey here with the video lecture on Chapter 14. The previous video lecture that I had loaded into D2L on microbiology comes from the old textbook. I think there are some good hints in there, some of the ways that the mnemonics, tricks that I use to remember which antibiotics have what mode of activity, so that might help you. This one will follow the order of our current textbook, the OpenStax textbook, and cover some of the bits from that. A lot of the material is similar, but our OpenStax book goes into a little bit more of the history and just has a different order, but much of the information similar. So either one or both of these videos should be very helpful. So first we'll look at the history of chemotherapy and the discovery of antimicrobials. This is not something new. We tend to think of it as something that's very new, but skeletal remains from Nubia, which is the area in southern Egypt and northern Sudan, has found traces of tetracycline in skeletal remains. Tetracyclines are a naturally derived antibiotic. They come from Streptomyces bacteria, and those Streptomyces produce this antibacterial as a way to reduce competition from other bacteria. So they outcompete their neighbors or others trying to gain a foothold in the same area by producing an antibacterial that will prevent the growth of these other bacteria. So tetracycline was actually found in the skeletal remains, and it turns out they were actually brewing. So when we brew alcohol, we use fermentation process, and there are bacteria that ferment and fungus, and they were using Streptomyces to brew their beer, and it looks like it was pretty intentionally used as a medicinal treatment. And going back to Indian and Chinese herbalists, back to thousands of years, they've used fungus and plants with antimicrobial properties in teas, in poultices, ways to prevent the growth of microbes, fungus and bacteria, and other pathogens. So historically, in these ancient times, they used the complete organism, the complete fungus or plant. And now a big area of biomedical research is finding out exactly what component of this plant or animal is responsible for these antimicrobial properties, so that we can extract just specifically that compound. And when you know what that compound is, we can actually synthesize it in the lab. So we can get a synthetic version or man-made version of it, as well as natural versions. So look at the first antimicrobial drugs in recent history. Paul Ehrlich was a physician. He was looking for a magic bullet for a drug that could cure syphilis. And his assistant, Sahachiro Hata, discovered what was named compound 606, the 606th compound that they attempted in 1909. And this turned out to be the magic bullet that was effective against syphilis. And that was marketed under the name Salvarsan. And it's still, we still use the method that they followed, which was just go through this vast array of here are all the possible things we want to sample. We're going to try each one until we find one that does the job. So a synthetic antimicrobial. So the term antibiotic specifically refers to compounds that prevent the growth of bacteria that are derived from naturally existing sources. So typically they're produced by other bacteria or by fungus. Synthetic antimicrobials we typically call chemotherapeutic agents. So when we think of chemotherapy, we tend to think of cancer, but that term chemotherapeutic is any chemical compound that's synthesized in the lab that we use to control disease. So synthetic antimicrobials are those that are man-made. They are developed from chemicals. They're not found in nature, but a lot of times the chemicals that are used to synthesize them, we figure out based on what is being produced by some natural product. Sulfanilamide was the first in a family of sulfa drugs that are still very widely used today. That was the first synthetic antimicrobial. And it was developed from a dye that was first used to treat patients in 1933. So this dye, when patients metabolize it, when they break it down, one of the intermediate products of that breakdown process, so one of the metabolites is has antimicrobial properties. So we still use these. An example, when you think like, oh, a dye, why did they think to do that? You've used this in the lab. When we use eosin methylene blue and McConkey's agar. McConkey's agar has carbon fuchsil in it, and that inhibits the growth of gram-positives. So we can still use stains and dyes as a way to inhibit growth. We do that in a lot of our selective media. So the first natural antibiotic, these are drugs that are produced by microbes, and they kill or inhibit other microbes. And Alexander Fleming is the researcher who is credited with discovering this. Florian Chain, two other researchers, had also kind of discovered all about the same time, and they came up with a way that they could scale up the production of this to make it happen at a scale needed to make this commercially available. But Alexander Fleming discovered penicillin quite by accident when he left plates growing in his lab in the incubator, and when he returned, found that there was fungal growth from penicillium on the plate, and it had inhibited the growth of the bacteria. So semi-synthetic antimicrobials, we see semi-synthetic, they are derived from the natural antibiotic products, so from antibiotics that are derived or produced by bacteria, but they have been chemically modified. So a portion of them is modified or synthesized in the lab and connected to a naturally occurring compound. And we do this, we modify those naturally occurring compounds to give them characteristics that are desirable. Sometimes it's to increase their stability. If we need them to remain active in the body, for the longer they're active in the body, the more effective they can be. So we don't want them to break down as quickly, so we can give them some increased stability so they can act for a longer period of time. We might be able to broaden the types of microbes that they can target. So they may be targeting gram-positives, and we can amend them in the lab so that they can also affect gram-negatives or vice versa. Or sometimes they can be toxic to our own cells. A lot of these are have toxic effects on the kidneys, and so we want to reduce their toxicity. All right, so some of the fundamentals when we talk about antimicrobial chemotherapy. We've already covered this. We talked about this in our previous chapter on controlling growth. Bacteriostatic versus bactericidal. So static, we're going to inhibit the growth. They cannot increase in number. So we stop binary fission, we stop their replication. Bactericidal kills microbes, and the way we define microbial death is a permanent lack of reproductive ability. So even if I remove them from the agent that is inhibiting their growth and I give them their optimal conditions, they're still not going to replicate. They're still not going to grow. That's the criteria that's used to define microbial death. And that's how we determine if something's bactericidal. And we'll come back to that when we look at ways to test the effectiveness of microbes. And then our usage, whether we select a bacteriostatic or bactericidal compound, depends on the patient status. So what is it that we're trying to treat? Oftentimes, so traditionally, bacteriostatic, if we can kill it, that's good. We want to kill it. But that can also drive a lot of antibiotic resistance. So now there's more of a lean toward bactericidal and let our own immune systems, bacteriostatic, let them stop growing so our own immune system can take over and eliminate them. Versus bactericidal. In cases where a patient is immunocompromised or it's an extremely aggressive pathogen, then we probably want bactericidal. So it depends on individual situations what we want to select. When we look at drugs, we have narrow spectrum drugs. These have a narrow range of action. They have a limited target of microbes that they can act on. And broad spectrum, they'll pretty much kill any microbe. We prefer, what we want to do selecting drugs is narrow spectrum. We want to target it as much as possible so that we can target the pathogen without killing our own normal microbiota. In this case, you have to have an idea of what the cause of infection is. If we aren't sure, if you don't know what the cause of infection is, if it could be multiple species infection, could be more than one bacteria and maybe more than one type, then we might have to go with broad spectrum. So for mixed infections, or we don't know what's causing this, but it's aggressive, we may go with broad spectrum. The downside of broad spectrum antibiotics or drugs is that they can kill our normal microbiota, and that leads to superinfections. So superinfection is when we have an overgrowth of bad bacteria because we've gotten rid of our good normal microbiota that keep the bad guys in check. So when we talk about this spectrum of activity, narrow spectrum typically means, oh, this targets gram positives, or this targets specifically gram negatives, or this targets specifically mycobacterium, whereas broad spectrums are gram positive, gram negative, everybody. So having a targeted antibiotic is preferable when that's possible. So you mentioned superinfections. Superinfections are secondary infections. It's typically not the pathogen of interest, the pathogen that's causing the disease, but it's often just an opportunistic pathogen that's present anyway, but its numbers are kept in check by our own normal microbiota. We take a broad spectrum or inappropriately targeted antibiotic, it can kill all the cells that are not resistant, including our normal microbiota that would be keeping this pathogen here in check, this opportunistic pathogen. And when we kill those normal microbiota, it gives our drug-resistant pathogen an opportunity to flourish. So when we talk about biota, we talk about our normal microbial colonists. These are the ones that our healthy bodies have everywhere. They are typically harmless or beneficial bacteria, and they keep the bad guys in check. A few of these normal biota may be pathogens. A certain percentage, about 3 to 5% of the population, has methicillin-resistant Staphylococcus aureus as part of their normal microbiota, but as long as it's in balance, they are not causing disease in these people, or it's part of their normal biota. Broad spectrum antimicrobials, destroy those healthy pathogens along with the one that we are trying to get rid of that's causing the disease. And so any of those pathogenic organisms, the opportunistic pathogens that are part of the normal resident biota or that are present but unable to multiply because of our normal biota, once we kill those with antibiotics, then those pathogens are able to increase in number. And some common examples of superinfections, these are commonly seen in the healthcare industry, are urinary tract infections caused by E. coli. So typically lactobacilli that are part of the normal flora in the female vagina are killed by broad-spectrum cephalosporins that are commonly used to treat UTIs, and they result in an overgrowth of Candida albicans, our number one cause of yeast infections, of fungal infections. So that's a really common side effect of taking antibiotics. There's also antibiotic-associated colitis. So a lot of antibiotics, tetracyclines, clindamycin, and other broad-spectrum penicillins, synthetic and semi-synthetic and natural, kill off normal biota that are found in the colon, resulting in an overgrowth of Clostridium difficile. And this bacteria releases toxins and cause horrible symptoms, gastroenteritis, diarrhea, fever, abdominal pain, and they're very difficult to eliminate. So those are some common superinfections where treating whatever was causing the initial infection ends up causing a superinfection of a secondary pathogen. Right, so some other considerations when we're looking at what antibiotic to prescribe or what chemotherapeutic agent to prescribe. Once we figure out, well, exactly what the pathogen is, what are we treating? Do we need a broad spectrum? Can we target it with a narrow spectrum? Do we need bactericidal or bacteriostatic based on the condition of the patient? We also have to look at dosage and route of administration. So typically for children, we're going to base the dosage on the mass, on body mass, base it on the drug metabolism. So what are the intermediate products produced by this drug as it breaks down over time in the system? A lot of these, the intermediate products can be toxic. So depending on the condition of the patient, we need to consider that. Patient status as far as immune system function, allergies, are they allergic to penicillin? Then we can't prescribe that, even if it might be our preferred drug. And the half-life of the drug, because, of course, the longer it is present, the longer it can act to control the growth of the bacteria. So what is the half-life? Is it long enough? How long should we continue to administer it? How long will it stay in the system? And then the route of administration. Typically, oral drugs are preferred. Again, the status of the patient must be considered. So not conscious, might not be able to administer oral drugs. Some conditions like dementia, are they going to be able to remember to take their medications, to take them accordingly, or do we need some other way to administer those? And then plasma levels in the patient. Also have to consider when we look at patient status, what other drugs are they taking? We can end up with drug interactions. Sometimes we want interactions. Sometimes we have synergistic interactions. That's when two drugs used together are more effective than either one alone. For example, trimethoprim and methoxazole. A lot of these are tongue twisters. So just hint, some of you are probably even more familiar with some of these names than I am. I go to YouTube and I type in, how do you pronounce, and whatever. And there are pronunciation keys. So if you are looking at these and the tongue twisters and want to use that as a source, it's very helpful. So trimethoprim and sulfamethoxazole. Together, these two are more effective than either one alone, and a lot of times that allows the dosage to be reduced. So if either one of those has high toxicity, having them together, we can reduce the one with the higher toxicity. And that's also a way to eliminate antibiotic or reduce antibiotic resistance, because by alternating or by having multiples, we can prevent any population of bacteria that might be resistant to one. They're probably not going to be resistant to both. So we can kill populations that have some resistance present by having multiple or combinations of drugs that work together. I also have to be careful about antagonistic interactions when two drugs used together, or a drug is prescribed while the patient's also taking some other drug, where they are less effective than one drug alone. An example of an antagonistic interaction is that taking antacids can negatively affect a lot of antimicrobials by lowering stomach acidity, raising the pH in the stomach, lowering the acidity. If a drug is absorbed in the stomach through that process, it needs a specific acidity for that to happen. So have to know not only how drugs will interact with each other, but what is the patient taking? What other medications are they taking, including over the counter? All right, so exactly how do antimicrobials work? Well, antibiotics have specific targets, and these targets for antibiotics are bacterial structures that are different from the structures found on eukaryotic cells. This is called selective toxicity. We want to find parts of the target cell or prokaryote that can be attacked without that same drug attacking our cells. So there are a lot of parts that cells have that all cells have in common. So we have to look at what's different so that these antibiotics don't attack our cells. So selective toxicity is the ability of the drug to kill or inhibit microbes while causing minimal or no harm to the host. So we look for targets in Chapter 3, all of those structures that bacteria have, compare those to the structures that eukaryotes have. Where are they different? What's different in the two? Things that bacteria have that eukaryotes don't have make good targets. And so the mode of action is going to be targeted to the cellular level of bacteria, to some part of their cellular structure or function that differs from eukaryotes. So the mode of action, or the targets that we look for in antibacterials, some common ones, those that inhibit cell wall biosynthesis. They look at the production of peptidoglycan. So our gram-positives in particular have to keep producing peptidoglycan. Every time they grow and divide, they need to build more cell wall components. So this is a very highly metabolically active ongoing process, particularly when we're in that steep exponential growth phase of the growth curve, the logarithmic growth. Every time those bacteria divide, they need to build more peptidoglycan. We don't have peptidoglycan. We don't have cell walls on our cells, so this is a really good target. And because it's being produced a lot, it's energetically intensive and it's going on all the time, that makes it a good target. So if we can inhibit the biosynthesis of proteins is another way that we can prevent that. So we can inhibit cell wall biosynthesis. So if we stop production of the cell wall, components of the cell wall of peptidoglycan, when the bacteria divides, it doesn't have that wall protecting it. And antibiotics can get in. We can subject it to osmotic lysis, so that cell wall's protection is gone. Another target is protein biosynthesis. The ribosomes, all cells have ribosomes, but the size and shape of the ribosomes are slightly different. So eukaryotes have 80S ribosomes, prokaryotes have 70S ribosomes. The shape, and I know this is weird because the sum total of the subunits of the ribosomes in prokaryotes is 80, but they have 70S is the grand total when these units come together. The smaller subunit and the larger subunit come together and we get a 70S ribosome, which is different than eukaryotes. So we can target that because it's different. So we can make an antibiotic that recognizes those differences, that it's able to act on the prokaryotic ribosome, but it can't act on the eukaryotic because it's a different shape and size. So by targeting that, we target the biosynthesis of proteins. If you can't make proteins, you lose your metabolism. Some protein synthesis, those proteins are used for structure, membrane proteins for all sorts of different functions, but most are going to be produced as enzymes that have a specific role in metabolism, carrying out some chemical reaction that the cell needs to survive. So if we in some way affect that enzyme, then that step in a metabolic process doesn't get done. And metabolism, have to have metabolism. We need the products we need from those cellular reactions, from those chemical reactions in the cell when we need them. So those ribosomes and protein synthesis are a really good target. We can disrupt cell membrane. So this one, I think, well, cell membranes. We have to look for things that are different in cell membranes. We always have that phospholipid bilayer. So that one's a tough one. We have to look for what's different between our membrane and the plasma membrane of bacteria. Well, the one good membrane target would be that lipopolysaccharide outer layer, in gram-negative bacteria, because our cells don't have that. It is a phospholipid layer, but it's structurally different with those polysaccharides embedded. And we can look for specific targets, membrane components, those different membrane components, the proteins that are on the membrane that make our bacterial membranes different from our own. Another target is DNA replication or transcription. So if we can inhibit nucleic acid synthesis, we keep those DNAs, the DNA molecule, the bacterial chromosome, from replicating or from being used as a template to transcribe RNA, so we don't get any RNA. Without either one of those, the cell can't function. Without DNA replication, cells can't divide. If they can't divide, they can't increase in number. If we can't build new DNA, we don't get new cells. If we can't transcribe RNA, then we don't get cell function, because that RNA is then going to be used as the instructions for how to build proteins. And again, we get to those enzymes that are responsible for all the chemical reactions in a cell. No chemical reactions, no metabolism, and the cell will die. What we can target, well, we replicate our DNA and we transcribe our DNA into RNA. Luckily, though, in order to do that, even though we use similar enzymes, DNA polymerase, RNA polymerase, those polymerases, those enzymes, are different for bacteria than they are for eukaryotes. And so we can target the enzymes that are responsible for these processes to prevent them from happening. We can target specific metabolic processes. So we can target the enzymes themselves, and that could stop a metabolic process. We can target production of the proteins, which would prevent us from having the protein that'll be the enzyme that'll have the metabolic process. Or we can go right to the process itself, and we can interfere with the expression of or with that protein's function. So we stop the metabolic process. This is usually a competitive inhibitor. So antimetabolites are antibiotics that are going to target some part of the metabolic process by stopping the activity of the enzyme. So the rest of the process, the DNA transcribed RNA, the RNA went to the protein, the enzyme was built. But if we can make a competitive inhibitor, a molecule that is similar in size and shape to the enzyme substrate, to the product or to the reactant in the chemical reaction that this enzyme facilitates, competitive inhibitor will land on the enzyme's active site and prevent it from working. Then we disrupt whatever that process is. Some examples, folic acid synthesis is a really common one because we don't make folic acid. We have to get that from our diet. So we can interfere with this and it will have no effect on us. It's in the mycolic acid synthesis enzyme. So the enzyme that builds mycolic acid in Mycobacterium, we don't make that product. So we can interfere with this enzyme safely for our cells because we don't have that enzyme. We can also target the production of ATP. This is another one that targets Mycobacterium. So Mycobacterial ATP synthase inhibitor. We're going to inhibit the enzyme that synthesizes ATP in the Mycobacterium. This is part of the process for how a cell makes its ATP that it then uses for energy. So if we can block the enzyme that's used to build ATP from adenosine diphosphate and phosphate, then the cell has no energy source and it can't carry out any functions. So these are all targets here. This the ATP synthase in bacteria and Mycobacterium is different from what we have. So some of the cytochrome C, some of the molecules involved in that process, the ATP synthase, are different enough that it affects them, but not us. So let's look more specifically at which classes of drugs do these. So inhibitors of cell wall biosynthesis. These are particularly effective against gram-positives because they are constantly building more peptidoglycan. So some of these can be used against gram-negatives. Those tend to be synthetic or semi-synthetic. We've had to alter the mode of action so that it's effective. These are typically beta-lactams. What beta-lactams, this class of drugs that are all similar to penicillin. Penicillin was the original. They have this beta-lactam ring here. So all of them have this similar structure. This is naturally occurring penicillin. And then these are man-made, derived synthetic and semi-synthetic varieties that have been altered. That main structure, the basic structure, has been altered in some way to give it specific properties. So our beta-lactams are penicillin and other antibiotics that work similar. They attack the cell walls, cephalosporins, monobactams, and carbapenems. These all have this same central structure. And what they do is they block the cross-linking of the peptide chains in peptidoglycan. So if you remember, our peptidoglycan, we have like long repeating units of NAG and NAM sugars. that give us these big, long, solid bars that are connected in layers and connected to each other, side by side and in layers, with peptide chains, with small 4-amino acid chains that link them together. This prevents those peptide chains from forming, and so those sugars aren't connected to each other. So it'd be like having a chain link fence where the chains don't crisscross. So all of these act in the same way. Look at the spectrum of activity. Penicillin, primarily gram-positive, a few gram-negatives. Cephalosporin, similar. Ampicillin, get a few more gram-negatives, but primarily, again, gram-positives, gram-positives, gram-positives only for carbapenems. So our penicillins are natural and some are semi-synthetic. They are very selectively toxic to gram-positives. The semi-synthetics can kill some gram-negatives, but the naturally occurring ones are primarily gram-positives. Allergies are pretty common, and so we have to look for alternatives. And sometimes those semi-synthetics or synthetics are good alternatives. So, for example, cephalosporins are good for patients who do have penicillin allergies. These are semi-synthetics, or synthetics have been developed. Monobactams are the group that have a very narrow spectrum for use against gram-negative. This is the exception to these being primarily used for gram-positive. Carbapenems have the broadest spectrum, gram-positive and negative. Glycopeptides like vancomycin are very narrow spectrum and used primarily for the treatment of multi-drug resistant gram-positives. So this vancomycin is often prescribed for MRSA, for methicillin-resistant Staphylococcus aureus. That MRSA, don't let the term methicillin-resistant fool you. Methicillin is the strongest drug that they're resistant to, pretty much everything else as well. So vancomycin is the one thing they're susceptible for. And now we're developing Versa strains of Streptococcus aureus, vancomycin resistant, which are resistant to all the things MRSA are and vancomycin. So bacitracin, this is a topical antibiotic. Typically, we're looking at oral antibiotics used for internal treatment, but this is for systemic. Bacitracin is typically or most commonly used for skin infections on the surface because it's nephrotoxic if used internally. It's toxic to the kidneys. This is effective in staph and strep infections, so gram-positives. And bacitracin ointment, when we talk about that, that's part of a triple antibiotic treatment. So then the groups of drugs that inhibit protein biosynthesis by targeting the ribosome. So we have two subunits to the ribosome, the smaller 30S subunit, and then the larger subunit on top, the 50S, that has the sites of action in protein synthesis. The A, the attachment site. This is where the transfer RNA comes in and attaches. Its anticodon pairs up with the codon on messenger RNA, carrying its amino acid. Then it translocates, it moves to the P, to the peptide site, where we form a peptide bond between the amino acid that just came in and the next one at this stage. And then we translocate, we move to the next stage of on the MRA, to the E site, the exit site, where I'm going the wrong way backwards. This is the A site. I usually draw it the reverse way, where the tRNA comes in, attaches with its amino acid, moves to the P site, where a peptide bond is formed between these two amino acids, and then the E, the exit site, where the transfer RNA leaves, leaving behind this growing polypeptide chain. So here our ribosome is moving this way to the left. down reading this messenger RNA, the codons. So we can disrupt, because these are different size from eukaryotic ribosomes, we can disrupt their action. We have antibiotics that specifically target this lower, the 30S subunit. So aminoglycosides, things like streptomycin, gentamicin, and neomycin, are going to prevent or impair the proofreading. So they prevent the proofreading, the anticodon and the codon, from matching up correctly. And that leads to errors. So they might, rather than pair up with the codon, they might shift by one nitrogenous base or just read incorrectly, maybe move over two bases, whatever their triplet reading is affected. And so they don't match up the right anticodon with the correct codon, and we get the wrong amino acid in there. And then that stops protein synthesis, the elongation of this peptide. We end up with shortened peptides. And they are able to, if this is a protein that's going to be part of the plasma membrane, one of the proteins in that membrane, it can disrupt membrane function. Tetracyclines also act on the 30S subunit, and they prevent the transfer RNA from binding here. If transfer RNA doesn't come in, then we don't get any amino acids in this chain, and we don't have the protein that it was going to build. We have some antibiotics that act on the 50S subunit, so this upper part here. These are things like erythromycin, chloramphenicol, which is very toxic, so we don't use that too often, azithromycin, and linezolid. I think we talked about these already on here. So aminoglycosides, these can be nephrotoxic, neurotoxic, ototoxic. They can cause deafness. They can cause hearing impairment. They're broad spectrum and bactericidal. Streptomycin was the first drug that was effectively used as a treatment for tuberculosis. First broadly used antibiotic that was widely distributed. Tetracyclines, also broad spectrum, and these are bacteriostatic. These were also discovered in the 40s. They have some side effects like phototoxicity, hepatotoxicity, so liver damage, and permanent discoloration of teeth. Erythromycin, this 50S unit, some of these prevent this larger unit from binding with the smaller unit, so they prevent the formation of the entire ribosome unit. Those two subunits are in the cytoplasm separate until messenger RNA comes along, and then they join. And then some just prevent the messenger RNA from feeding. They block this site where the messenger RNA feeds through. All right, so erythromycin is one of those. This was discovered in the 50s. It's bacteriostatic, and there's lots of semi-synthetics available. Azithromycin is a broader spectrum. It has fewer side effects, can be used for a much shorter period of time. So everybody's kind of familiar with the Z-Pak. This is pretty common now, where you can have a reduced use, reduced dosage. So shorter period of time, fewer number of doses to take. Chloramphenicol. This was the first broad spectrum drug to be approved by the FDA in 1949. It's a natural antibiotic, but can be produced synthetically very easily and cheaply. But it has a really serious side effect. It suppresses bone marrow production of blood cells. Can also cause aplastic anemia. So it's very rare that it's used in humans today, but many animals treat it or tolerate it better, don't have those side effects, and so it's still used in veterinary medicine. And then linezolids are broad spectrum. They are synthetic antimicrobials, and these target that 50S subunit, prevent the messenger RNA from feeding through. All right, our next mode of action, inhibitors of membrane function. So polymyxins. Polymyxin B is a narrow spectrum, specifically targets gram-negative bacteria, that outer polysaccharide layer. So it interacts with that LPS, with that the outer membrane, and eventually by disrupting the outer membrane, because we have that very thin layer of peptidoglycan on gram-negatives, when you disrupt that outer membrane, it's attached to that peptidoglycan. It kind of breaks that apart as well, so then it can get to the inner membrane, but it has to go through the outer membrane first. It's narrow spectrum, primarily against gram-negative. Its toxicity restricts its use to topical use, primarily topical use. This is also part of that triple antibiotic ointment. It's another variety of polymyxin. Colistin targets gram-negatives and also multi-drug resistant strains of bacteria. Its toxicity restricts it to only using those multi-drug resistant infections to kill normal flora before intestinal surgery. So we limit that because it is very toxic, and because it's because it's not targeted very well. It does kill the normal flora, so we use it for that in cases where we want to eliminate all bacteria, all potential for bacterial infection by opportunistic pathogens like before surgery. And then daptomycin targets gram-positives, and it's also used for multi-drug resistant strains. This has less toxicity, so can be administered through IV. Membrane. There we go. Nucleic acid synthesis. So here, when we're looking at nucleic acid synthesis, primarily we're targeting the enzymes that carry these processes out. We're not targeting the DNA specifically, because our DNA and bacterial DNA made of the same stuff. But how we copy it, the enzymes we use to copy it or to transcribe it, are different. So rifamycin targets bacterial RNA polymerase, so it stops transcription. So RNA polymerase, that's the enzyme that builds the polymer RNA. We stop that. We don't have the message for how to build the protein, and the proteins don't get built. So we're going after the construction or the synthesis of RNA polymerase, or after the RNA polymerase itself, rather, after that molecule, so we don't get transcription. This is part of a combination therapy that's used for tuberculosis. So this targets RNA polymerase. Fluoroquinolones inhibit bacterial DNA helicase. So helicase, remember, is the enzyme that unwinds and unzips our double-stranded DNA. So it unwinds it and unzips it. We also call this, in bacteria, we call this gyrase. So gyrase. This gyrase is different from the helicase in eukaryotic cells. So we can target this specific enzyme. If we don't unwind and unzip our two strands of DNA from each other, then DNA polymerase can't access the template strands in order to build their corresponding complementary strands, and we don't get DNA synthesis or replication. If we don't replicate our DNA, our bacteria don't divide, and that's the end of the population. This is broad spectrum. It will cover gram-positives, negatives, mycobacterium, but its toxicities limit its use to much more serious infections. Right. If we look at the antibiotics that inhibit metabolic pathways, what we look for are metabolic pathways, so things that bacteria build or break down that we don't. Remember, metabolism, the sum total of all the chemical reactions in a cell. Those chemical reactions can be anabolic, building big molecules from small ones, or catabolic, breaking down big molecules into smaller ones. All of those are assisted by an enzyme. And typically we go through a metabolic pathway, so there are multiple steps to get from our starting product to our end product. Each one is facilitated or catalyzed with an enzyme. And so we look for metabolic processes, chemical reactions that bacteria have that we don't. And then we target those metabolic processes by stopping the enzymes that carry those metabolic processes out. So the sulfa drugs, sulfonamides, are the big class of these. Trimethoprim, isoniazid. I know I'm saying that one wrong. I just looked that up. Can't remember how to say it. I can never remember how to say that one. Isoniazid. You can look that up. You might know how to pronounce it. You can tell me. All right, so our sulfa drugs block the synthesis of folic acid. Folic acid is essential in the production of nucleotides, our building blocks for DNA and RNA. We have to get folic acid through the foods we eat, but bacteria can make that. So that is not a metabolic pathway we have, so we can use that as a target. And these are used in the treatment of leprosy. But allergic reactions to sulfa drugs are common. Trimethoprim also targets folic acid synthesis. Isoniazid blocks mycolic acid synthesis. So we can use this in the treatment of tuberculosis and leprosy. Remember our Mycobacterium, Mycobacterium tuberculosis, Mycobacterium leprosai. Those are the bacterial species that are responsible for these. Our Mycobacterium have mycolic acid. Remember, they were the gram-positives that we have to have a special staining technique for. We have to use the acid-fast technique because this mycolic acid, this waxy layer on the outside, makes it impossible for our gram staining process to reach the peptidoglycan that it reacts with. So we have a special process. So this targets the synthesis of that mycolic acid. Without that protective outer waxy layer, that bacteria is more susceptible to destruction. And our ATP synthase, another pathway, we can block ATP synthase in Mycobacterium. This has really serious side effects, so it's typically only used for untreatable cases of tuberculosis. Right, so those were all targeting bacterial or prokaryotic structures and functions that differ from our own cells. But we also have some other microbes that are eukaryotes. So what do? So for antifungals, we can go after cell membrane disruption. When we disrupt the membrane, whether it's on a bacteria or a eukaryote, what we do is we end up allowing ions and chemicals that should be on the inside. We let them leak out, and we let chemicals that are on the outside that should stay outside enter the cell. Basically, without the cell membrane, the cell falls apart and all the insides fall out. So we can disrupt the membrane on fungus because the sterol that's part of their membrane is different from ours. We have cholesterol in our membranes that is used to maintain the consistency of our membrane. Fungus have ergosterol. So we can target that ergosterol in the plasma membrane. We can inhibit nucleic acid synthesis. Again, we have similar enzymes involved in these processes, DNA polymerase and RNA polymerase, but they're slightly different versions of it, so we can target that. Fungus have a cell wall. Again, we don't have a cell wall. Theirs contains chitin, so we can target chitin with those, or we can disrupt the microtubules, which are different from what our own cytoskeleton microtubules. So we can target those, are all different. Miconazole, this, if you see azole at the end of the name, probably an antifungal. These treat fungal skin infections and yeast infections by targeting ergosterol in that plasma membrane. Nystatin treats yeast infections of skin and mucous membranes and fungal infections in the intestine, again, targeting that membrane sterol. We can treat systemic fungal infections, so these are internal. Amphotericin B, again, ergosterol. It's a good target for us. Capsifungin goes after that cell wall made of chitin, and this is used to treat aspergillosis. This is the most common cause of asthma, is aspergillosis. It's when we get the spores from aspergillus in the lungs, and then that can grow. It's dimorphic, so we can get it as a single cell spore, and then it can grow into a multicellular mass in the lung. It's also used to treat systemic yeast infections. Yeah, you'll have to figure out how to pronounce this one. Graciofulvin. Graciofulvin. I have to look that one up. So this inhibits the microtubules, and again, it's oral for skin infections, fungal skin infections. When we look at drugs to treat protozoans, again, harder to come up with selective toxicity with protozoans because they are eukaryotic cells. They have lots of different modes of action. Chloroquine is used to treat malaria. Pentamidine is used to treat African sleeping sickness. Here we go, Metro. Metro is used to treat pretty much, if you have to guess what it is, it's treating a protist infection, the antiprotozoal. This is the one that everybody gets, is Metro. Metronidazole. Metronidazole. Something like that. And we've got lots of different modes. They can inhibit electron transport chain in the mitochondria of the protozoan. They can inhibit folic acid synthesis, so some protists are able to do this, and again, we don't. They can produce damaging reactive oxygen species. So some of these, we have lots of intermediates from cellular respiration that are toxic, and if we have those metabolic pathways for aerobic cellular respiration, we have evolved enzymes to break down, to take care of those toxic intermediates. So if we can produce a toxic intermediate an organism doesn't have an enzyme to take care of, we can use that as long as it's something that we have the enzymes to take care of. We can inhibit DNA synthesis, again, slightly different versions of DNA polymerase and RNA polymerase, or inhibit heme detoxification, so blood group detoxification. We break down and release those ions. And then we have a group of antihelminthic drugs. Ivermectin is really commonly used in veterinary. If you have dog or cat, you've probably had ivermectin at some point. Niclosamide treats tapeworms. Praziquantel is used for tapeworms and flukes. You can get this over the counter. So a lot of the things to treat worms, we can get dewormers over the counter. Can inhibit microtubule formation, reduce glucose uptake. Without glucose uptake, we can't go through cellular respiration to get ATP. Block nerve transmissions and cause paralysis. can inhibit ATP production by targeting specific proteins in the mitochondrial membrane that are involved in the electron transport chain, induce calcium influx, or inhibit RNA synthesis. Again, different enzymes. Our antivirals. So antivirals, now we're going to things that have to be inside a host to replicate and spread. So it's really hard to treat because we have to put those drugs into our cells because that's where those viruses are replicating. So to stop that replication. So some of the drugs, some of the ways we treat these, acyclovir is a common antiviral. It's used to treat the herpes class of viruses, so herpes, chickenpox, Epstein-Barr, cytomegalovirus. It halts elongation in DNA replication. So it stops us from making a copy of DNA. And the way it does that is this is a man-made analog of guanosine. So that's my nucleoside. That's the deoxyribose, the sugar that's found in DNA, and guanine. So it looks similar to this, but not quite, but similar enough that it gets incorporated into the building strand, the new strand that's being built of DNA, but it's different enough that it doesn't work and it prevents that DNA from actually rejoining and forming a double helix. Antiretroviral therapy. It's used in the treatment of HIV. That's our retrovirus that carries reverse transcriptase. And so we have an inhibitor, AZT, that this inhibits the function of that enzyme of reverse transcriptase that prevents the virus from reverse transcribing DNA from its RNA. It also includes protease inhibitors. So protease are enzymes that build proteins. So we stop building the proteins for the capsid. Integrase inhibitors that prevent the DNA from being incorporated into the host DNA, and fusion inhibitors that prevent the virus from actually attaching to the host cell to begin with. So it's this combination. And then we have some other antivirals that are used for some common viral treatment. I'll let you read through those. So drug resistance. So I really want to go back. Drug resistance, this understanding how this happens really depends on understanding what was going on in chapters 9 and 10 with DNA, with microbial genetics, and with genetics and understanding how natural selection works. So drug resistance is an adaptive response in which microorganisms begin to tolerate an amount of drug that would normally be inhibitory. It is not, we don't have the ability to alter our DNA, our genes, our genetic makeup. Right? Our genes are our genes are our genes. The way they are altered is through mutation. So this adaptation in biology means, hey, somebody has the right genetics that allow them to somehow tolerate this amount of a drug that would normally inhibit or kill them. So it's due to genetic versatility and adaptability. Adaptability happens over time, over generations, as we pass on mutations and the numbers with that changed genome increase. I can't say, oh, I want to be resistant to this antibiotic. Here, look, this antibiotic's all around me. Let me change my genes so I'm resistant. If a bacteria is not resistant, if it doesn't have the genetics that make it resistant to the antibiotic, then when the antibiotic is introduced, it will be killed. So the resistance already has to be there. The genetic change that allows it to tolerate the antibiotic has to be present in the bacteria. A mutation had to have occurred prior to exposure to the antibiotic. Antibiotics don't cause the mutation. Antibiotics don't change the genetics. The bacteria don't intentionally change to become resistant. Within the population, there's variation. There's variation because of horizontal gene transfer. There's variation typically because of binary fission, because that is cloning. There's very little variation other than what arises through this natural background mutation rate. Some mutations are lethal. They don't get passed on. Some are not lethal, and then they do get passed on, but at very small numbers in the entire population of bacteria. The way the numbers of that mutation, so that mutation is neither beneficial nor harmful, is how it stays in the population, but it has to arise first. When the environment changes, meaning when the antibiotic is introduced, those that have that adaptation will survive, and they will pass it on to their offspring. Those that don't have it will not survive. So in the next generation, the bacteria are going to have that mutation, and then they will replicate, and that will continue to be passed on as part of the growing population. It will increase in number. Initially, though, because only a few will have that, most of the population will be killed off. So we think, oh, the antibiotic worked. But then as that population that got reduced down to just the few that had the mutation, that starts to grow because it no longer has any competition, increase in numbers. Oh, all of a sudden I feel sick again. Let me take that same antibiotic. Now that population all have that mutation and are resistant, and it doesn't work. That's how drug resistance or antibiotic resistance comes about. So it can be intrinsic as well as acquired. Intrinsic means it's there. Some of the bacteria have this mutation, this genetic makeup. It's not giving them any benefit, but it's also not harming them. Or it can be acquired, and those are those horizontal gene transfer methods that we talked about. So resistance to penicillin developed as early as 1940, but it was still relatively rare. But then over time, as it became less expensive, easier to get a hold of, to get prescribed, penicillin expanded its use to everything, weren't very selective, just gave out penicillin like candy, so that by the 80s and 90s, scientists began to see treatment failures on a large scale. And microbes were becoming newly resistant, either because of a spontaneous mutation in some critical gene, or acquisition of new genes through horizontal transfer. Chromosomal drug resistance usually results from a spontaneous random mutation. It can be a very slight change. So if you remember the activity where we saw sickle cell anemia, the chain of amino acids, that's what our DNA tells us, how to build this polypeptide that will become a protein that will have a specific function. And to go from normal shaped hemoglobin that is very adept, it is perfectly designed to carry lots of oxygen, and it's perfectly designed so when I pack a red blood cell full of hemoglobin, I get a nice round red blood cell that tumbles and flows nicely. But in sickle cell anemia, my hemoglobin's misshapen, it can't carry as much oxygen, and when I fill a red blood cell with it, I get a misshapen red blood cell, all because one base pair in one triplet was substituted. So that's the power of a mutation at a single base pair, a single point on DNA. So that one mutation can change the shape of a protein to the point that, in that case it was something harmful, but it might be changed the shape of a protein. For example, the protein on the cell surface that the pathogen has to bind with to gain access to the cell. I change the shape of that protein by just a little bit, and that pathogen can no longer gain access. So it can be a very slight change. We can get slight changes in drug sensitivity with larger doses. So sometimes it's just a matter of that small change can prevent it a little bit, can hold off on it. So if I just take a bigger dose, I can overcome it. That's not always the case, but sometimes. Or if I just continue to take the drug for long enough, I will overcome that slight bit of resistance. So that's why it's important to take the complete dosage for the complete length of time, the entire treatment that's prescribed. Persisters are bacteria that have very slow metabolism or are able to stop their metabolism. So as long as that antibiotic is present in the system, they can stop being metabolic, they can slow their metabolism and not be affected. Well, eventually you stop taking the course of the antibiotic or it breaks down, and then they increase their metabolism and so they won't be harmed. So that is another way that organisms can be resistant to the effects of the drug. We can get resistance through horizontal transfer. So we can transfer resistance factors, the portion of the chromosome or a plasmid, extra chromosomal piece of DNA that's carried by the bacteria that's resistant, that confers that resistance, and it can be transferred through transformation or transduction or conjugation. There are plasmids that are encoded with drug resistance that are naturally present in microbes. That must be present before the bacteria is exposed to the antibiotic, and then it can be passed on. Transposons can actually duplicate, insert genes for drug resistance into plasmids, and then we can just spread those through those horizontal gene transfer methods. So this is a great image of how we get this spread of antibiotic resistant, the increase in numbers. So I have my broth or my agar or my plate that doesn't have any antibiotic in it. And all these bacteria are growing. I have all these different colonies. And let's see, a mutation, drug-resistant mutant arises here and here, and those colonies begin to grow. Well, as those colonies grow, there's still way more bacteria that are not drug-resistant because they're all competing for the same resources. They have no advantage. Their numbers are not going to increase over the numbers of the other bacteria. But when I add the antibiotic, those that don't have that resistant gene are all wiped out. Now, all of these non-resistant species or non-resistant colonies are no longer competing for the resources on this plate. These guys are still replicating. They are still able to undergo binary fission because they are resistant to the effects of the antibiotic that's on the plate. And so they pass that mutation in their DNA down to their offspring, who replicate and pass it on and replicate and pass it on. So that ends up with the dominant species present being all resistant to that bacteria. So some of the mechanisms that bacteria use to become resistant, beta-lactamases are enzymes that break that beta-lactam, that central component of penicillin. If they break that down, so beta-lactamase, they break that down, the antibiotic no longer prevents them from building cell wall. They can block the entry of a drug into a cell. So we can actually just change this entry point so that protein on the membrane that the antibiotic enters through can be blocked. We can do target modification or drug modification, so that would be changing, either inactivating or changing the drug. So beta-lactamases change the beta-lactam ring on the penicillin. They change the drug. Efflux pumping, this is common again among gram-negatives. They actively pump the antibiotic out. So if you can actively pump that antibiotic out, you have the ability to do that, to make a surface protein that is a pump specific. It's a channel protein that's specific to the shape of these antibiotics. You can get rid of it. Target modification, you change the target so the antibiotic doesn't work. So you have some slight change to your ribosome or some slight change to your DNA polymerase or your RNA polymerase. And so the target is no longer the shape that the antibiotic is designed to act on. The CDC issued a threat report in 2013 that outlines potentially catastrophic antibiotic resistance. So we may be entering a new post-antibiotic era where there are some infections that are completely untreatable. So now there's a lot of research into new ways to attack bacteria, new ways to treat that are not antibiotics, or ways that bacteria are unable to develop resistance to that treatment. So, and it's hard to bring new antibiotics to market because they're not an economically lucrative market. They're time consuming to develop. So we don't have a lot of new ones coming. So looking at alternative ways to treat antibiotic resistance. Some of the threats that the CDC report specifically addressed were Clostridium difficile, carbapenem-resistant enterobacteria. So these are gut bacteria that are resistant, and drug-resistant gonorrhea. There are some other serious threats, and then a lot of multi-drug resistant, and our streps and staphs that are vancomycin resistant. So if we look at MRSA, MRSA is a widespread opportunistic pathogen. VRSA, or our vancomycin-resistant enterococci, and VRSA, the resistant Staph aureus. This is a resistant species, prevents vancomycin from binding. Carbapenem-resistant gram-negatives use lots of different mechanisms. So those are just some examples of growing concerns. If you're going into a healthcare field, those are going to be more and more widely seen, bigger threats that I have to come up with new treatments for in the future. All right, last portion of this, how we test the effectiveness of antibiotic. How do we know what antibiotic to prescribe? How do we know it's effective? How do we know when it starts to cease being effective? So the Kirby-Bauer disc diffusion method, this is something that's typically done in lab. I think you have a virtual lab that shows this. You use a specific agar called Mueller-Hinton, and where the agar is made to have a known diffusion rate of antibiotic through there so that I can compare the size of the zone of inhibition. From one lab to the next, if everybody used Mueller-Hinton, and the same dosage on their discs, this is standardized so I can compare those zones of inhibition. So we place filter discs with the antimicrobial compound and a specific dosage, specific amount here, after we've laid a lawn of the bacteria. So the bacteria, this plate's inoculated by taking a sterile swab and then just spreading that sample everywhere. Then we put these filter discs on the plate and we culture it. If the organism's able to grow right up to the disc, like here, then this antibiotic's not effective against whatever this organism is. So it'd be one organism per plate. The zone of inhibition and the size of the zone of inhibition indicate if the organism is susceptible. So here, whatever this drug is, the organism that I inoculated this plate with is susceptible to it. Here, the organism's resistant. I interpret by measuring how effective this is. I measure the diameter of the zone of inhibition, and I compare this to an antibiogram, which is a table that tells me how how big my zone of inhibition has to be to determine if my organism is susceptible to this drug or if it's resistant to the drug. So this is another example where I would just measure. Here is my diameter. I go and compare. These are my discs. I'm not sure if you saw this in lab where it tells me what the antibiotic is, tells me what the dosage is, and then I measure and compare to my table. All right. A broth dilution test is another way to determine the susceptibility. In broth dilution, I have a serial dilution, but what I'm diluting is the dosage. So the concentration of my antimicrobial. And what I'm looking for is the minimal inhibitory concentration. That's the lowest concentration that inhibits growth. I can also determine the minimal bactericidal concentration from this. So here I have growth, right? My tube's cloudy. So this I know, the 8 micrograms per mil is my minimal inhibitory concentration. I know bacterial growth was inhibited at this concentration because I have no cloudiness. I do not know by looking if it's bactericidal. So I have to do a second step. So my tube dilution test is more sensitive and quantitative than the Kirby-Bauer test. I serial dilute that antimicrobial in the broth, in each broth. I inoculate, I incubate. The smallest concentration of drug that visibly inhibits growth is my minimum inhibitory concentration. To determine my minimum bactericidal concentration, my MBC, what I would do is find my minimal inhibitory concentration, and then I would transfer a sample of this. I know there's no growth in this tube. I'm going to transfer it to fresh media that doesn't have the inhibitory agent, that doesn't have the antimicrobial, and I'm going to give it its optimal conditions and incubate and grow. If it grows, then I know that concentration was only bacteriostatic. It was the minimum inhibitory concentration. If the bacteria don't grow even under their optimal conditions, then I know it was that's the minimal bactericidal concentration. And sometimes I might have inhibited at 1, so I would test all of the subsequent dilutions because it might be inhibitory here, but bactericidal at a little higher concentration. I can also do this in a microarray. So rather than an entire test tube, I can just inoculate these tiny tubes. Same thing, a series of dilutions. Look for growth. Where I have no growth, the first well with no growth tells me my minimal inhibitory concentration. And then I would subsample all of these concentrations onto optimal conditions to see if that was my minimal bactericidal concentration or just inhibitory, and where my minimal bactericidal concentration is at. All right, I know this is a lot. Highly recommend you watch my other video. It's shorter than this one from the old textbook because I give you some ways to remember which of the drugs act on what. Primary focus is on the antibiotics, the broader, the antifungals, just know broadly, know generally. Same thing with the antiprotozoans and the antihelminthics. All right. I will get chapter 15 up sometime this weekend. Thank you all. Have a good weekend.